So today I will be talking about primary congenital hypothyroidism. To start, I'd like to describe what the thyroid gland is and its function. It is a small butterfly-shaped gland at the front of the neck. You can see here in this diagram. It has receptors all over the body, but the thyroid hormone functions really to boost metabolism and growth. So the receptors on the somatotropes of the pars distalis and the anterior pituitary, when they're stimulated by the thyroid hormone binding to its receptor, it will stimulate growth hormone synthesis and further body growth. Thyroid hormone receptors on the heart, when they're stimulated, will increase heart rate, contractibility, and cardiac output. And thyroid hormone receptors on skeletal muscle and adipocytes, which are fat cells, will increase basal metabolic rate. So primary congenital hypothyroidism is considered when there are low thyroid hormones, so T3 and T4. There are different types. So thyroid dysgenesis is when the thyroid gland itself does not properly form, it's absent, or it is in the wrong location. Thyroid dyshormonogenesis, which I will be discussing today, is when the thyroid hormones themselves are not being produced correctly. And so as a result of these low thyroid hormones, an individual will experience a slower metabolism and slower growth. Some of these symptoms as a baby can be shown as slower than normal. The baby is sleeping more. Dry skin, constipation, weight gain, muscle weakness are all also symptoms. An intolerance to the cold is also a symptom because with lower thyroid hormones, we'll see a lower body temperature. And then long-term effects of primary congenital hypothyroidism can lead to intellectual disability and short stature. So the molecular cause of primary congenital hypothyroidism is a missense mutation in the solute carrier family 5-member 5 gene. So this SLC5A5 gene codes for the sodium iodide symporter. So the NIS lays on the basal membrane of the follicular cells within the thyroid gland. The NIS functions to bring iodide and sodium into the cell so that these iodides can bind to tyrosine, and that can form thyroid hormones. So if the NIS is not properly formed, then no iodide can be brought up into the cell, and therefore thyroid hormones cannot be produced. So the mode of inheritance of this disorder is autosomal recessive. So I'd like to kind of go over some of the vocabulary related to inheritance. One of those being heterozygous and homozygous. So as a heterozygous carrier, you have a dominant and recessive allele. So even though an individual does not have the disease, they do carry a recessive allele that can be inherited to their offspring. And then with a homozygous individual, they have two of the same allele. So they either are dominant, homozygous, and they do not have this disease, or recessive homozygous, where they have two recessive alleles, and therefore they carry the disease. So autosomal recessive, you need to inherit two recessive alleles to develop this disorder. These Punnett squares can show some of the probability of inheriting primary congenital hypothyroidism as an autosomal recessive disorder. Here we see that a capital T indicates a properly functioning NIS. This is a dominant allele. And then the lowercase t represents a recessive allele as the improperly functioning sodium iodide symporter. So on this first Punnett square, we see two parents that are dominant for a properly functioning NIS. In that case, there is zero probability that their offspring will develop this disorder. In the next Punnett square, we see that there is one parent that is afflicted with this disorder. They have two recessive alleles, but the other parent is dominant for a properly functioning NIS. So there is no probability of an offspring developing the disease. In this third Punnett square, we see a parent afflicted with this disorder, and then the other parent does not have the disorder but is a heterozygous carrier. So they do have one recessive allele. In this case, there is a probability of 1/2 that the offspring will develop the disease. In this fourth Punnett square, we see that both parents have the disease, and so there is also a probability of 1 because the offspring will only have the chance to inherit a recessive allele. So we can see this autosomal recessive inheritance pattern in pedigrees as well. So we can see here that there is the parent that has the disorder. The other is dominant for the functioning NIS. But because the parent that has two dominant alleles will give the dominant allele to each of their offspring, none of the offspring will show that they have the disease. None of them will develop it. But they are going to become carriers because of their parent that is afflicted with this disorder and can only pass on recessive alleles. So we can see here that this carrier did marry a person afflicted with this disorder, and there we see that the disease has passed on to one of the offspring. In this area, we see two carriers that have 4 offspring. One of the offspring has developed its disorder because of the 25% chance here of developing the disease. And then we see another situation of 2 carriers having offspring where 2 of them have been afflicted with this disease. So on to the genetic testing of the SLC5A5. This gene is analyzed through the Baby Genes Targeted Panel. It is a newborn genetic test that uses buccal swab, cord blood, isolated DNA, or whole blood to analyze the SLC5A5 gene. It is costly, around $800, and it is optional. And I think it's mainly optional because of the newborn screening that is mandatory in the United States. This newborn screening measures thyroid stimulating hormone and thyroxine levels in the infant's blood. This is measured within 2 to 4 days of life. This is mandatory. It is measured in such a short amount of time of life because of how important early detection is. If left untreated for more than 2 weeks, low thyroid hormones can lead to permanent effects. And typically, we cannot detect primary congenital hypothyroidism without newborn screening. So we can see the importance of this early detection in this graph here. On the x-axis, we see the chronological age, and then on the y, we see developmental age. The red line shows normal development, but these green lines here show children that are thyroid deficient. So here, even after thyroid treatment, we do see a small increase in bone age and height age, but not to the level of normal. And here we see that the mental age, even with thyroxine treatment, does not improve. And this just shows how important it is to detect this disorder and provide the proper treatment.